have Dr. Anne Marie Wills, who is the director of the CurePSP Center of Care at Mass General Hospital, which provides multidisciplinary care for people with atypical Parkinsonisms. She's the principal investigator, or PI, of the CurePSP Genetics Program, and additionally the co-PI of the multicenter Progressive Supranuclear Palsy clinical trial platform, which is known as PTP. Her research focuses on genetic and environmental risk factors for neurodegenerative diseases, particularly PSP and Parkinson's. Chinyere is a clinical research coordinator at Massachusetts General Hospital. His primary focus is on the genetics of movement disorders, and he's been a rock star with crunching all the data and making sure everything goes really smoothly. So with that, I'm going to pass it over to the two of you to talk to everyone today.
Great. Thank you, Courtney. Just as a quick introduction, I just want to let everybody know, unfortunately, Sonia Schultz, who is an important collaborator on this project, is not able to join us and participate in this webinar because of the government shutdown. So the NIH has been shut down and she is not allowed to do any work on this or any other programs. And so she's not allowed to even join this webinar today. So I'm going to be trying to sort of stand in for her portion as well. And I apologize that I may not be as good as Sonia. But I'm going to go ahead and start sharing my slides now. And Courtney, please let me know if this, if you can see this. Does this look good, Chinyere? Is this
great?
Okay. We thought it would be a really good time. It's been exactly 1 year since we launched this project. So we thought it would be a really good time to give everybody sort of an update on how things are going. I think things are really exciting. And we do have, you know, some information to share with you today. Right, let me just flip. So I'm just going to start out with like a little bit of the background of PSP, CBS, and MSA in terms of our genetic understanding, what we know about them so far. Then we're going to talk about sort of how the program has been going, the specific results from our genetic project, and then we'll sort of wrap up with the questions at the end. So I just want to emphasize that most cases of PSP, CBS, and MSA do appear to be sporadic. So meaning that there is no family history when people get the disease. There are several case series that suggest that maybe 15 or 20% of people who have these diseases might actually have a family history of a similar disease. In the case of PSP, maybe 15% if you include PSP and Parkinsonism in prior studies. In MSA, it looks like a little higher, 20% of Parkinsonism alone in people who have a diagnosis of MSA. So, but the vast majority of people do not have a positive family history. So maybe 85% of people don't have a family history, and we consider them sporadic. We'll say if you do have a positive family history, we'll call them familial. There have been case reports of individual families over time in the literature. This is just a summary of most of the families that have been reported in the PSP literature. And this is, you can see sort of the family trees over here on the left. We'll talk a bit more about family trees later in this discussion. And here, everybody who has PSP is shown in black, and you can see that there are individuals sort of dotted around some of these families. Some of these families look like there are more affected individuals than others. But unfortunately, nobody actually identified what the underlying genes were in these families. And these cases were reported so long ago that we actually cannot get the DNA from these families anymore. But this is something that I think is really interesting. And one of the reasons why we wanted to do this study was also to try to find families with these diseases who are still living, who could participate in genetic research. This is just sort of a global view of our understanding of neurodegeneration so far. So as you can see, diseases like Alzheimer's and Parkinson's, where there are a lot of people who have these diseases, we've been able to identify a lot of genes that underlie these diseases. And that has actually been really helpful in terms of trying to come up with treatments for these diseases. Over here on the far bottom left corner are the diseases that we work on, PSP and CBD and MSA. And unfortunately, as you can see, we don't know a lot about the underlying genetic causes of these diseases. And in part, that is because of the low frequency in the population. So I'm going to talk a bit about some of the sort of more sort of classic Mendelian genetics underlying PSP now. So one of the questions actually that was submitted in before the webinar asked about the H1 haplotype. And so I wanted to sort of explain that a little bit for people because I think a lot of people have heard of it. Basically, what a haplotype is when you have genetic markers that show that certain genes cluster together and are inherited together. So this haplotype, the H1 and the H2 haplotype, they include the MAPT gene, but they also include other genes around it, and they all sort of move as a block. You know, you inherit a whole block of genes from one parent or another. And it's true that the H1 haplotype is associated with higher gene expression of MAPT, meaning that more tau protein is produced if you have an H1 haplotype. And it is also true that people with PSP are very, very often carriers of the H1 haplotype. So here on the right is just an example of a cohort which was sequenced, and you can see that most people had the H1 haplotype, very few had H2. But I just want to emphasize the H1 haplotype is the dominant haplotype, meaning most people in the world have the H1. Meaning that even if you have the H1 haplotype, it does not mean that you're going to get PSP. It seems to be a risk factor, but it's definitely not enough to give you the disease. So I just want to reassure people about that. But it also suggests that there is some sort of subtlety within the H1 haplotype that we don't understand, how it works, how it affects, for example, the tau protein or what it's exactly doing. But there is definitely a signal there. So in general, you know, we talk a lot about MAPT and PSP. This is the gene that we know the most about. It's the one where if you have a mutation, it is likely to give you one of these diseases. CBD also, you can see MAPT mutations or C9orf72 mutations. It is quite rare, though, to see these mutations. And in MSA, people have seen mutations in GBA and synuclein. These are genes that are classically associated with Parkinson's. We know a lot about them because of the Parkinson's literature. And interestingly, those genes are genes that are being targeted in clinical trials. So I think that is sort of the hope for why we're doing this type of research, is that it will lead to a future treatment. This is work that Dr. Schultz did, where she took several hundred people with sporadic MSA, and she looked at their genes across the whole genome. You can see these little peaks here, signals that suggest that there is some marker that increases your risk of getting MSA. We don't really know what that risk could be. We know that, you know, there's some sort of variation going on, but it doesn't necessarily mean that there's a mutation in this gene. It could be farther downstream of this marker. It could be that this mutation is affecting, you know, transcription elsewhere in the genome. We don't actually know what this means, but it does suggest that there are some risk factors for MSA that we really don't understand or know about. And so hopefully, as we increase the numbers of people and their genes and their DNA, it'll help us to identify what these underlying causes are. This is from a PSP GWAS. So this is actually almost 3,000 people with PSP, and again, sporadic PSP, people who gave their DNA for research, both in the US and in Europe. And so sort of combining all of them into one big study, identified again MAPT, but also some other peaks near these other genes. Again, that doesn't necessarily mean that these genes are the culprits. It could be something nearby. But we don't actually know sort of how this is going to translate into getting the disease. But it does suggest that there could be some genetic factors that affect even people who have sporadic forms of the disease. I just want to go back in time. Quite a long time ago, the MAPT gene was first identified, and it was identified because of one family that seemed to have a very strong risk for getting a Parkinson's dementia sort of complex. So you can see here, this is a really sort of the classic sort of Mendelian inheritance pattern where, half of the children in each generation are inheriting a gene and then getting the disease. This does suggest, that it is a highly penetrant gene, where everybody in black here is affected. The MAPT then was associated with PSP because people were able to find mutations in that gene in individuals with PSP and CBS. And interestingly, you can see how all of the mutations seem to cluster around exon 10 here. And that means that this is just sort of a general slide on sort of how do mutations lead to disease. Well, so if you have a mutation that changes, for example, the structure, the folding of a protein, it can affect that protein's function and how it interacts with other proteins. And so in the case of MAPT, those mutations do affect how tau binds to microtubules down here. So it prevents it, for example, from binding and stabilizing microtubules. And then the tau can aggregate and form the classic inclusions that we see under the microscope in PSP. So how often do you see MAPT mutations in PSP? It is extremely, extremely rare. So if you take everybody, all comers, it's less than 1% of people will have a MAPT mutation. And it's reported to affect up to 11% of people with familial cases. Actually, we think that it's actually lower than that, perhaps as much as 5% at most. But just to reconfirm, there's a lot of people who don't have these mutations who are getting PSP, and we really want to try to figure out why. It is important also to remember that genes are just part of the picture. It doesn't explain everything about why we get these diseases. There's definitely some environmental risk factors, and my team is actually working on that as well. And then there's going to be interactions between genetic risks and environmental factors that could impact people getting these diseases. So it's obviously extremely complicated, but we do need to really try to dig into both of these areas to try to figure out what causes these diseases. So the question is, how can we find out more about the genetics of these diseases? And the answer is with your help. So with a huge collaboration between our center and CurePSP and the NIH and GP2, which is the Global Parkinson's Genetics Program, and Indiana. We've been working on this CurePSP Genetics Program for the last year. We're going to talk about the results in a second. I just want to sort of explain a little bit about what's going on with the NIH shutdown. So because the NIH is really intrinsic, is really central to the DNA analysis and sequencing, we've had to put a pause on enrollment during the shutdown. So we have put in a protocol amendment to our IRB to try to come up with a workaround, which I'll talk about in a second. But I just want to emphasize, we are pausing the sample collection and the shipping to the NIH, but we are still able to take in your information. And as soon as we're able to collect your blood again, your DNA again, we will be able to resume sample collection and the DNA processing. But so just hopefully this is temporary. We don't know how long the shutdown is going to take. We have put in an amendment to try to bypass the NIH. So this would mean that the samples would come directly to us at MGH, and then we would send the samples to a GP2 affiliated laboratory, so a different laboratory for the sequencing processing. But we don't know how long that's going to take to get the amendment through, and we don't know how long the shutdown is going to take. So we're going to see, we're going to try to, we don't know. Hopefully they'll open up. Hopefully this amendment will go through. Hopefully we'll be able to resume, all the activities within a month. But just to let people know sort of what's going on, and just sort of to editorialize for a second, I just want to emphasize, people often say, oh, we don't really need the government to do things for us. This is an example of how useful the government really is. The NIH really is doing important work and doing important research, and we really do need the NIH to come back online. So I'm going to pass it on to Chinyere now. He's going to talk about the results.
Hello, everyone. My name is Chinyere Obasi. I'm one of the study coordinators for the CurePSP Genetics Program here at Mass General Hospital. I'm going to spend some time kind of keep getting you up to date about how the study has been going, some more recent results, and stuff moving forward. Now, as you know, the program launched on October 4th, 2024, and we've seen consistent enrollment in the study since then. We had a really big spike in that first month in October, 70 registrations in the first month, more than 2 a day. And then we received another big spike in January, February, when CurePSP started putting out advertising on the study with greater intensity. In the first 12 months, we've received nearly 500 registrants, and we were able to consent and enroll 265 of them by the government shutdown on October 1st. And as you can see from the graph, we've seen an increase, a big uptick both in October and also for this year, but it's been pretty consistent in enrollment ever since. We are seeing a slight slowdown, but even without intense advertising or being put in too many public information, we're still getting people enrolling nearly every day. As you know, for the study mainly works on travel lab phlebotomy appointments. That's at-home phlebotomy appointments run by phlebotomists in your area. Of the 265 consented and enrolled participants, 211 of them have been able to have successful travel appointments in their home for a home blood draw, proving how well this system can work moving forward. We have had the occasional unsuccessful appointment, but in most of those cases, we've been able to either draw it again later on or have them go to a local clinic for their appointment. We've had the occasional patient no-show or cancellation. This is usually due to miscommunication between the participant and the phlebotomist. So we do ask that if you're in the study, when the phlebotomist reaches out to you, please get back to them as soon as possible. And then we've had 12 appointments not go through due to a lack of geographic coverage. What this means is that there was not a travel lab phlebotomist in the participant's area. This has primarily been an issue in New Mexico and in parts of the rural Northeast. Like I said before, we've been able to get most of these through local clinics, participants going to their local hospital or PCP and having their blood drawn there. But moving forward, we also have a plan to add additional local labs, contract with local labs, in order to allow participants who might live in an area with less coverage the opportunity to participate in the study more easily. On screen now is a study flow diagram of the study as it exists right now. As a kind of a snapshot from October 1st, like I said before, 490 participants have been registered as of that date. We've had to exclude 154 of them for a variety of reasons. The primary reasons we've noticed are due to duplicate appointments. So people will sign up once and then we might take a little bit to get back to them and then sign up again. We do ask that if you sign up once for the study to not sign up again. We did receive your initial participation survey. We're just taking some time to get in contact with your doctor. In addition, we've had some issues of participants who have signed up, but then later on were unable to participate or withdrew consent. We've also had some signups we had to screen fail due to diagnosis. This is primarily in one of two cases. Sometimes participants who have a condition similar to PSP, but not quite PSP, CBS, or MSA will sign up for the study, and we have to screen fail them due to the fact that the study is specifically focused on those three conditions. In addition, we have occasionally had the healthy children or family members of people with these conditions sign up. We just want to let you know that we don't recommend healthy asymptomatic children or siblings of people with these conditions to get genetic testing for a variety of reasons, including the impact on your future insurance. We've had to screen fail a couple of participants due to geographic location, mostly Canadian participants. We apologize. And then at the very bottom, you can see some were simply unable to provide informed consent or weren't able to coordinate with their legally authorized representative for the consent process. An LAR, to be clear, is a person who helps you with the consent process from a legal perspective. This can be a healthcare proxy, a power of attorney, or a next of kin. So we do ask that if you are signing up for the study and you think you might need an LAR for the consent process, that you coordinate with that person before with that person to come to the meeting together to make the process go smoother. Of the 265 people enrolled and consented, the vast majority have been people with PSP, but we have had sizable minorities of people with CBS and MSA also enrolled, with numbers that are relatively equivalent to what we would expect from the national population. We've occasionally had discrepant diagnoses. For example, a person might sign up with one condition and their doctor might disagree, but the condition they disagree with is still a condition we are covering, and so we tend to err on the side of enrolling them if we have at least some evidence they probably have one of these three conditions. On the phlebotomy result at the very bottom, like I said before, 211 of these participants were able to have their blood draw done via travel lab, and an additional 10 were able to have their blood draw done via local lab. At the very bottom, you'll see a number of pending appointments, currently around 25. This number is slightly inflated due to the government shutdown. We haven't been able to do any blood draw appointments since October 1st. But we do ask that when the shutdown is over or when our workaround is properly approved, that you get in contact with us and answer our phone calls so we can get that pending appointment number down as fast as possible. This is a quick demographic snapshot of the study population as it currently exists. We have roughly equivalent participation of men and women in the study, with a slight increase in men as equivalent to our expectations based on the gender within these conditions. The average age of a participant in this study is 70.5 years old, but that is very variable. We've had some much younger and much older than that. In general, people have their consent meeting after being diagnosed with the condition for around 2 years, but that is also very variable. We've had a good number of participants sign up literally the day after they get diagnosed, and some towards later in the disease progression. In general, the participants in our study tend to be highly educated. The majority of them have at least a bachelor's degree. The past year has been wonderful for the study, but we have had some challenges and some lessons we've had to learn about how to move forward. A big one is that when we call participants, people often think we're spam or think we're trying to sell something. We understand this, especially today when there's so many telemarketers, we understand this impulse. We do ask that if you see a number calling from Mass General Hospital, specifically this number, the 617-643-2400 number, please answer. It is the study calling, probably about either a blood draw appointment or a consent appointment. We've had some issues obtaining diagnostic confirmation forms from treating neurologists. We understand this as well. We understand neurologists are very busy, but getting those forms filled out by your neurologist has been very important and also a bit slow at times. We've had occasional issues of participants being too ill to participate or having some technology challenges with Zoom. Again, we understand this. Zoom is not the easiest tech to work around. We have had a high percent of participants who require a legally authorized representative. Like we said, this is someone who is legally there to help you with the consent process, usually a healthcare proxy, a power of attorney, or a next of kin. So we do ask that if you sign up thinking you might need an LAR to consent into the study, or if we let you know that you need an LAR based on your doctor's note, that you coordinate with that person so that you both come to meeting at the same time and the whole process can go a little bit smoother. And then, like before, we've had some dependence on Travel Lab. We have had an impressive amount of geographic diversity with Travel Lab. It's been truly staggering, but we've had the occasional issue of Travel Lab not being in a specific area. And so we are, like we said before, working towards fixing that issue moving forward. On the diagnosis side, like we said before, the vast majority of participants enrolled in the study have been diagnosed with Progressive Supranuclear Palsy, but we have had sizable minorities with CBS or MSA, roughly equivalent to what we'd expect in the national population. Within PSP, the vast majority of participants have been diagnosed with probable PSP Richardson syndrome. Again, this makes sense. This is the most common presentation of PSP. That being said, we have had significant and interesting minorities of participants showing up with variant subtypes of PSP, most notably PSP Parkinsonism and PSP with predominant gait freezing in yellow and purple on the screen. Now we get to my favorite part of family history. Everyone in the study provides family history during the consent meeting. Of those who provided family history during the consent meeting, 3.8% of participants had at least one family member with PSP and also themselves had PSP. So 3.8% of the participants had a PSP family history. We don't just ask about PSP, we also ask about a variety of other neurodegenerative diseases. And when we include other conditions like ALS, Parkinson's, and FTD in our metrics, that number jumped to 24.5% of participants with PSP having at least one family member with a similar neurodegenerative condition. If we were to include Alzheimer's, that number would jump even higher. In general, the most common family history for PSP participants is one family member with Parkinson's, followed by PSP and ALS. And we define family member in this case as parents, children, siblings, aunts, uncles, and cousins. If a family history presents as particularly interesting, we tend to make a pedigree. That's a visual map of the conditions progression to a family, similar to a family tree, but focusing more on conditions. And the one presented here represents kind of yes, participant on the left, he had a brother with PSP, a father with Parkinson's, and a maternal uncle with Parkinson's as well. And then all of these maps, PSP is coded in blue. What you'll see from these maps is definitely some family history of interest in the condition, but it does seem to be not the most penetrant and have some variable expressivity. Penetrance refers to the intensity of a gene presentation in your body. For example, in the top left, because the father has Parkinson's and the mother does not, we would expect at least half the participants, half the children of that family to have the gene we're looking for, but only 2 of them have presented with PSP. This suggests that the gene we're looking for might not be as penetrant as genes might have studied in school. In addition, there does seem to be some variable expressivity. What that means is that even if you carry the gene, it might express differently from person to person. So if you look at the family at the bottom middle, you see 2 family members with PSP, and the third that seems presenting somewhat differently. That does not necessarily mean that's a different gene. It might just mean it's the same gene expressing in different manifestations from person to person. This kind of over what I said in the previous slide, but familial PSP, while still rare, might be more common than previously thought. All families seem to have low penetrance, assuming that this is a single gene metric and monogenic condition. There also might be some variable expressivity within that same gene in the same family, which explains why we have different conditions showing up among siblings in the study. I'm now going to pass it back off to Dr. Wills, who's going to explain what happens to your samples once you submit them to us. Thank you.
Great. Thank you, Chinyere. Okay, so after you give blood, it is shipped to the NIH, or would have been shipped to the NIH, where they're going to turn it into DNA. And then they put it through 2 different sort of processes. So the first one is called the neurobooster array. This is kind of a quick snapshot where they look at over 1 million different variants that are known to be related to neurodegenerative diseases, and including some mitochondrial variants. So the first 96 people who signed up for the study have had this done. It's been processed, but it hasn't been yet fully analyzed. And unfortunately, since Dr. Schultz's team is unable to physically go in to the NIH and access their computers, they're not actually able to finish processing and analyzing these results. Same thing for the subsequent 96 people who already have had their DNA extracted. We're still waiting for them to go through this process because of the shutdown. The first 96 people who went through who donated DNA have also been sequenced. So this is using an Illumina platform. The sequencing has been completed. The analysis has not. But we are hoping that, assuming that the shutdown does not last forever, we'll be able to get those results. And then if we do see a positive mutation, if we see something that we think is likely to be associated with your disease, we will then reach out to you, get a second confirmatory cheek swab sample, and then send that to a different confirmatory lab for confirmation testing so that we can then give you the results back. So the whole process of doing, the initial analysis and the sequencing and everything was supposed to take just 1 year. Unfortunately, it has been pushed back a bit by all the problems with the NIH and now the shutdown. So I apologize, things have been taking longer than we expected, but we still hope to have these results back to you within a year and a half of having provided your sample. I just wanted to give a shout out to our colleagues at GP2. This is an amazing program, which is, they are sequencing over 150,000 people with Parkinson's from all over the world. And their goal is also to enroll 5,000 people with PSP worldwide. So we are one of the sort of largest cohorts here in the US, but there are cohorts elsewhere in the world. There's a very large cohort in Taiwan, for example, and in India. And so, you know, we're hoping that this is going to, you know, really find new and exciting genetic risk factors for these diseases by really coming up with a very broad, you know, genetic pool. So this is something that is still ongoing. This is a slide from Sonia. So just to sort of give the overall sort of goals for this study. So first is, of course, to try to find mutations and variants that are risk factors for these diseases. And then once we have some target genes to actually build models, be it in vitro or in vivo models of these diseases, and then using that, we can identify potential targets. So we can identify drugs that might interact with some of the genes that have been identified. And this process has worked, or is in the we are, for example, in Parkinson's disease, there are genetic targeted therapies for you know, synuclein and GBA that are in the clinic, in clinical trials right now. In PSP, we are targeting MAPT because that's the one that we know the most about. So the platform trial, for example, that we've been talking about, that is mostly aimed at the tau protein and trying to come up with ways of modifying the tau protein. But I think that it is important to try to find new and different and other types of targets for this gene, because we know that you know, this is not, we're only sort of scratching the surface right now in terms of what the underlying genetic structure is for these diseases. And this is just to sort of reiterate that the goal is really to come up with some targeted therapies that would treat the people who have known genetic mutations that lead to these diseases. So we do feel that we are on target. We know that, you know, obviously the NIH shutdown has affected things a little bit, but, you know, we've got 221 DNA samples that are currently being processed right now. And we are optimistic. We're still hoping to enroll 1,000 people within 3 years of starting this study. And so obviously we need as many people as we can to sign up and participate. We just want to, this is just to reiterate, we really need large numbers of participants to find new disease risk factors. And so we have a couple ways that you can sign up. The website is definitely the most important way. So this is if you go to the CurePSP Genetics Program here, website, you can sign up. And then, as Chinyere said, we will be reaching out to you or to your physician first, actually, to get their confirmation of your diagnosis, and then reaching out to you to do to enroll you, consent you, and take your family history. There will be hopefully the delay in terms of DNA collection and processing will not take too long, but the other portions of this study can continue. And this is the same page that we have for Spanish speakers. If Spanish is your primary language, we can also enroll you. And I want to thank everybody who's put in effort into getting this study up and running. This is the steering committee who helped in the design of the study. Obviously, all of our collaborators who are listed here, our funders especially are listed down here. This is our team here at MGH. The CurePSP team who's been working on this with us, the NIH team, and the GP2 team who's been helping us as well. Finally, the genetic counselors at Indiana University who hopefully will be able to reach out to you as soon as we are over this shutdown. So I think that's everything.
Great. Thank you so much, both of you, for that. That was extremely informative. And I learned, honestly, a lot about genes. Thank you. I have a bunch of great questions from the chat and the Q&A function, and I'm just going to start with some logistical questions about the program, then we can dive more into the science. The first one is somebody asked if the diagnostic confirmation needs to be from a neurologist or if it could be from a PSP, excuse me, PCP, not PSP, which is a primary care provider for everyone.
Right. So we would like it to be from a neurologist. Obviously, if you don't have a neurologist, if you were ever seen in the past by a neurologist, we would take their note as a confirmation. The way we set up the study was that ideally we have a confirmation form that's filled out by your treating neurologist. If we cannot get them or another neurologist who has seen you in the past to fill out that form, then we will accept, for example, a note, some sort of history. If you can send that to us, then we can fill out, sort of the details that we need to confirm your diagnosis. But yes, we would prefer the diagnosis to come from a neurologist. Yes.
Okay. And just to clarify a question, we're taking people for PSP, CBD, and MSA. I just saw someone ask if it was just PSP, but wanted to confirm that. The second question is, can family members without a PSP, CBD, MSA diagnosis get tested in this program?
So generally, no. If you don't have a diagnosis, we would not recommend any sort of pre-symptomatic testing. As Chinyere said, we worry that it could impact your future insurance, not necessarily your health insurance, but your disability insurance, your long-term care insurance, things like that. So we don't recommend that you get tested if you are asymptomatic. Now, we would envision in a few cases, for example, the families that Chinyere showed, in a few cases, it might be helpful to get samples from other people in the same family. So we do keep that open as a possibility. But in terms of the actual, we would be telling you if we want you to reach out to your family members to ask them to enroll. At this point, if you have a family member or a parent or something who had one of these diseases, we would not recommend that you sign up.
Great, thanks for that clarification. Okay, so the next question, this is a pretty popular question, is what do you recommend for those who have submitted a blood sample but not heard back? Is there a confirmation they should expect to confirm that the sample was received by the facility?
Yes, apologies. So we've been thinking about sort of how to make this happen in a more systematic way. So everybody who is given a blood sample at this point can rest assured that it was received and it was processed and their DNA was good. We haven't had any problems with the samples. We haven't had any sort of quantity not sufficient or, that the DNA came degraded or anything like that. All of the DNA has been great so far. So if you don't hear from us and you've given your DNA, you can rest assured that it worked, that it is in the process of all of that sequencing and analysis that I mentioned. And I didn't go through sort of the details of sort of how the analysis works, but there's a lot of computational work that has to go in the back end for every sample to try to figure out sort of whether, if we have, there's 3 million variants, right, that we each carry, right? And so trying to figure out which of those are relevant or pathogenic or stuff takes a lot of work. So apologies for the delay in terms of getting back to people about their results. We had initially anticipated that if it took, a year and a half and you didn't hear from us, it meant that you didn't have any mutations. It looks like because of the delays, it's going to take probably 2 years for everything to be fully analyzed and for us to be able to get back to you if you do have a mutation. So, but for now, rest assured that your DNA is good and it is being processed.
Okay, good to know. Thanks for answering that. The next question is, do people get a copy of the results even if there is not a variant found
So
or
if
mutation?
we so just to let you know, the first steps that I've been talking about are all for research, and so we cannot give you those results back. The only results that we can give you back are after we do confirmation testing. So that would be the cheek swab that we send to a different laboratory. So we're
Got it.
not actually allowed to give you a negative result. But yes, if we do find something and we confirm it with a separate lab, we will definitely give you those results, and we'll have a genetic counselor who can talk to you about the meaning of those results and how they might impact your family as well.
Mhm. And in this case, negative just means no variants were found, correct?
Right.
Okay, just wanted to clarify that. Okay, great. So let's move on to just some more questions specifically about the research and the results. Somebody asked if a brain autopsy shows if there are MAPT mutations.
Good question. Okay, so if you're participating in the brain bank donation program that CurePSP also runs, and your brain is going to Florida to the Mayo Clinic and being used for research, that the DNA from those brains is also being analyzed in a sort of parallel project. However, those results would not ever go back to the participant or their family. But yes, that DNA will be used for research.
Okay, great, good answer. Speaking of the brain donation, somebody had asked if you were able to accept blood samples from other samples that had already been taken and are in a lab, or samples from an autopsy.
That's a really great question. And it is really, really complicated. If there's a very, very strong family history, they should reach out to us and let us know. If this is just, a single isolated case, then I would say no.
Makes sense. Another question was, would any gene that is potentially responsible for a diagnosis be present from birth, or would it kind of progress over life? A mutation, I guess, is what that is referring
Yeah,
to.
so these would all be mutations that people would be born with. Yeah, so generally speaking, the same genes would be expressed throughout your body, the brain and the blood, and so you would be able to identify them. So these would be genes that you're born with, yes.
Great. So we had a question, recognizing that PSP is the focus of most research in the literature we have. And so people are just wondering if there are any words of hope or just ongoing research that you can talk about for CVD and MSA in addition to this program, which is totally understandable because it is.
Yes, yes. So I apologize that I had such a heavy PSP sort of presentation. Partly that's because this is sort of, sort of the biggest cohort of participants in our study. CBD A lot of the genetics looks very similar to PSP. So definitely, there's the same MAPT mutations, but there are also mutations in things like progranulin, TDP43, and other genes that have been identified in frontotemporal dementia. So there's some overlap there. And then MSA is very interesting. So we were talking about how there's a lot of overlap between genes for Parkinson's and MSA. There isn't as much research in MSA specifically, because of the low number of samples. So for example, the study that Sonia did, that I showed, sort of the genome-wide significance plots. So MSA, she had, I think, 600 and something samples, to look at, which is, not a lot. And so, we really do need to boost the numbers of people with MSA who participate in genetic research. Yeah.
Yeah, I'm grateful to those of you who are participating in this, which I think leads me to this next question. Was someone had said, if the goal is to ultimately develop treatments based on genetic mutations, how would the over 85% of patients with no known mutation benefit from this?
Great question. So this is definitely a challenge for the field. We are optimistic that, for example, in the case of tau, even though most people don't have tau mutations, the pathologic sort of appearance under the microscope looks the same. So you see tau aggregates under sporadic conditions, just like you do in people with, MAPT mutations. So we think that even if you don't have a specific mutation in one of these genes, the process and how you get the disease is probably related. And we would hope that we would be able to impact it in people with sporadic disease as well. But truthfully, we don't really know if this is going to work. I will mention, for example, as a sort of parallel example, in Parkinson's disease, there's a gene called LRRK2, which is relatively common. It's not as common as GBA, but it's like the second most common gene mutation that you can have in Parkinson's. And so there is a special drug that's being developed against that. And the company, like right now, there's a trial that's going on where the company's actually testing that drug in people who have just sporadic Parkinson's, so they don't have to have a mutation in that gene to be part of the trial. So they're testing to see whether it might actually impact everybody.
Got it. Sounds good. I'm gonna switch the gears just a little bit, and I keep writing questions in the chat and whatnot, folks. But someone had asked a question about, their grandmother had passed away from PSP, and I believe that her mother as well, if I'm reading that correctly. Do you have any recommendations on how, to educate themselves, or any advice if knowing that it's a potential genetic connection but no real answers? I hope that question makes sense.
Yeah, no, that's really tough. I mean, I think, one reassuring take home that I kind of wanted people to take away from this, from looking at like the families, the pedigrees that Chinyere showed, is that even if somebody in your family, like all the families that we've identified, even though there are families where there's, multiple people with PSP, for example, in that family, it doesn't guarantee that you're going to get it. Like most of the other siblings in those families don't have the disease. So I don't want people to be, too worried about their risk. It looks like a lot of these genes are of low penetrance. So hopefully that's reassuring.
Yeah, it's a good message of hope. We just got a question, and I'm curious if you are asking about veteran status in the application or eligibility criteria.
No, we're not.
Okay.
We do talk a little bit about a few risk factors in when we take sort of the history, but This particular study is not really looking at environmental risk.
That makes sense. Looking at the one that just most recently came through. Sorting through these. I just had one that I lost that I wanted to ask about. Going back to some of the logistics, to something you mentioned was to confirm, you are confirming research positives with patients who have submitted blood, correct? Okay.
Yes.
That's something just want to confirm that.
Yes. So if you have a positive like mutation and we think that it's related, then we will reach out to you to get a second sample. This would be a cheek swab sample. And then with that sample, we will have that, sequenced, and then we can give that back to you.
Yeah. And with the cheek swab, we actually got a question about that. It says if you've taken a blood sample and find the person would qualify for a mouth swab, but the patient has passed away, is there any alternative that could be provided, such as hair sample results on file with online heritage programs?
Okay, that's a really good question. So our workaround at this point is we do have a backup sample. So in this study, we've been sending, 2 tubes of blood to the NIH and 1 tube to NGH. So we do have sort of a backup saved sample here.
Great.
That will be our workaround.
Oh, awesome. That's great. And I think we have time for 1 or 2 more. And I wanted to just flag, we just talked about risk factors with veterans, and so I wanted you to clarify briefly what that meant for folks.
Well, so we think that, there could be some risks related to pesticide exposure. Interestingly, and this will be, I think, probably a future webinar. We've been looking at the UK Biobank to look at risk factors for PSP. The results are just, we're submitting them now. We were not actually able to confirm a risk factor for pesticide exposure, but the UK Biobank doesn't really capture that very well. We are obviously interested in trying to find more sort of risk factors for environmental risk factors for these diseases, but the challenge is that most studies are too small to have a large number of people with each of these diseases in them.
And then the last question we'll get to right now is someone is confirming that the cheek swab would still be 2 years from now, given the fact of the timing of the sequencing being completed.
I mean, so I'm hoping that the shutdown won't last that long. I would guesstimate that it might take another 6 months before we know, we're able to get that data out and analyzed. So I was being really conservative saying 2 years. Truthfully, we don't know how long the shutdown is going to take.
Great. Okay, well, thank you both for answering those questions. I think it was really helpful for folks, and I hope that if you haven't participated in the program, please do, with generous understanding of the government shutdown and all external factors. But I do want to flag in the chat, someone sent the clinicaltrials.gov. If you are interested in research studies, PSP, CBD, MSA, please visit there. If you have questions about that site, please feel free to reach out to us, but that's going to be the best place to go for any answers about available clinical trials. So thank you again for visiting today and joining us, and to those of you watching on YouTube, we so appreciate you taking the time out of your day. I keep saying it, but if you have any questions, please email us at info@curepsp.org. And it's not without all of your help that we can learn more about these diagnoses. And so we so appreciate everyone on the team and everyone who joined us today. Thank you all.